Good afternoon, ladies and gentlemen. Welcome to the Shaw Prize Lecture on Life Science and Medicine, 2017. I'm Justin Lee, MBBS Year 4 student of the Lee Ka-shing Faculty of Medicine. Today, we are most delighted to have this year's Shaw laureates, Professor Ian R. Gibbons and Professor Ronald D. Val, to share their insights and award-winning discovery. Your presence today will celebrate a huge leap in the field of life science and medicine. Professor Gibbons will share his discovery through a recorded video interview, after which Professor Val will share on the topic, Molecular Motors, the Engines of Life in person, and finally followed by a panel discussion. Before the lecture begins, may I invite Professor Peter Matheson, President and Vice-Chancellor of the University of Hong Kong, to deliver a welcoming address. Professor Matheson, please.
Thank you very much. Yes, so it's a great pleasure to welcome you to the 2017 Shaw Prize Lecture in Science and Medicine. It's a particular pleasure to welcome the laureates, one physically here, the other one you'll see in video, but represented by what was described in my speaking notes as his children. So we have two of his children sitting in the front row here. They may or may not like being described as children, but anyway, that's the way they're described in my speaking notes. Unfortunately, Dr. Gibbons himself is not well enough to travel to Hong Kong, and so you'll see him on video, and you'll hear a bit about his work, and you'll hear in person from Dr. Val. I was privileged to be at the Shaw Prize award ceremony last night, and if any of you have never been to the Shaw Prize award ceremony, I recommend going. It's sort of Hong Kong's equivalent of the Oscars, really. It's you have bright lights and music, and you have stages moving left to right. It's a really incredibly glitzy sort of show. But it's an illustration of the importance of the Shaw Prize in Hong Kong and in the wider world. And this is a very highly regarded set of prizes. The local universities get the opportunity to host a Shaw Prize lecture most years. Last year we hosted the Astronomy Prize, and I was saying to the laureates of this year's prize that I struggled to understand very much of the astronomy lecture, but I'm hoping to do better on microtubules because this is a field that I know a little bit about, and so I'm interested to hear an update from world leaders on this very important subject in cell biology. The Shaw Foundation has many links with the University of Hong Kong. We're very grateful to the Shaw Foundation, not just for the prize and for allowing us to host the prize lecture, but also for a number of other examples of philanthropic support from which we and other universities in Hong Kong benefit. I know there are a lot of school students in the audience, so I hope this is not your last visit to Hong Kong U. I hope you're going to come back as students, and then later on maybe as graduates and as teachers. So welcome everybody to the occasion. Professor Val, in particular, it's a great privilege to have you here to share your thoughts and your experience and your knowledge about microtubules. And I know that you've accepted the challenge of producing a lecture that will be accessible to the school students and also of interest to those people in the audience, of which I'm sure there are some who have their own expertise in the subject. A couple of things struck me about the acceptance speeches that were given last night by Professor Val and also by Ms. Gibbons on behalf of her father, and in particular the willingness to recognize the contribution of the other. I thought that was very striking. Professor Val even described Professor Gibbons as his hero, and I thought that was a very touching remark. So these were people who came together because of shared research interests, identified each other as being complementary in terms of the way that they could work together and push the field together in a very harmonious and very synchronous way. And so I think for the Shaw Foundation to award them together would seem to be a very appropriate and a very touching aspect of this year's prize. The other thing that struck me was the willingness of both Professor Gibbons and Professor Val to pay tribute to the students and to the other people that had helped them over the years. These prizes are always awarded to senior people, but the willingness to recognize that a lot of the hard work was actually done by much more junior people, many of whom may have moved on into other laboratories or other careers. That also struck me last night, and I thought that generosity of spirit and recognition was really very striking. So I'm looking forward to hearing more about the story. I'm hoping I'll be able to understand some of it. There'll be an opportunity for panel discussion and questions afterwards. But it remains just for me to thank you all for coming, to thank the Shaw Foundation again for the opportunity, and to reiterate my sincere congratulations both to Professor Val here, to Professor Gibbons in his absence, and to the friends and family that are with both of them. It's a great pleasure to have you all here at the University of Hong Kong. And with that, I'll pass over to the MC again to allow you to introduce the next stage. Thank you very much.
Thank you, Professor Matheson. Please come onto the stage. Sorry. As a token of our appreciation, we have prepared souvenirs for Professors Ian Gibbons and Ronald Val. Since Professor Ian Gibbons cannot attend the lecture in person, may I invite Professor Gibbons' families, Ms. Wendy Gibbons and Mr. Peter Gibbons, to receive the souvenir on behalf of him, and Professor Matheson to present the souvenir on behalf of the university. Please. Thank you, Professor Matheson. Please remain on stage. May I now invite Professor Matheson to present the souvenir to Professor Ronald Val on behalf of the university. Thank you, Professor Val and Professor Matheson. Please remain on stage. May I also invite Ms. Wendy Gibbons and Mr. Peter Gibbons, together with Professor Danny Chen, our moderator today, to take a group photo. Please. Thank you. Would Professor Chen please stay behind? Professor Chen, yeah. Other guests and professors, please be seated. May I now invite Professor Danny Chen, Assistant Dean, Research Postgraduate Studies of the Faculty, and S.Y. and H.Y. Chang Professor in Stem Cell Biology and Regenerative Medicine, to introduce the two Shaw laureates. Professor Chen, please.
Thank you. Professor Val, Professor Matheson, Professor Leung, distinguished guests, colleagues, students, ladies and gentlemen. Welcome to the Shaw Prize on Life Science and Medicine 2017. It is indeed my great pleasure and honor to introduce the two laureates for this year. I'll begin with Professor Ian Gibbons, who is not able to join us today, but as you know, he has recorded a message for us. He was born in Sussex, UK, graduated from the University of Cambridge with a Bachelor of Arts and then a PhD. He started his scientific career with a postdoctoral fellow at the University of Pennsylvania in 1957 and progressed to lecturer and assistant professor in the Department of Biology at Harvard University. Then to associate professor and professor of biophysics in the Pacific Biomedical Research Center, University of Hawaii, Honolulu. He retired in 1997 and moved to the University of California, Berkeley, where he has been working since as a research scientist and now a visiting researcher. Professor Gibbons is a fellow of the Royal Society and has received many awards, including the E.B. Wilson Medal, together with his colleague and wife, Barbara Gibbons, the International Prize for Biology, and of course the Shaw Prize this year. He and Barbara are well respected by their peers. Now, in a research presentation at a cell biology conference, where they show a fascinating video demonstrating the role of Dinant, a microtubule-associated motor protein, in the movement of sperm tails. At the end of that video, the audience was awed at their finding that they received a standing ovation. Now, not too many scientific presentations received that sort of recognition. The other laureate is, of course, Professor Ron Val, and we are very pleased that he's able to join us today in person. Born in California and graduated with a BA from UC Santa Barbara and a PhD from Stanford University. He started his scientific career as a staff fellow at the Laboratory of Neuroscience, NIH, and at the Marine Biological Laboratory at Woods Hole in 1985, and to full professor in UCSF in 1994. There he served as the director of the cell biology program and chair of the Department of Cellular and Molecular Pharmacology. In 1999, he was appointed an investigator of the Howard Hughes Medical Institute. Professor Val is a member of the U.S. National Academy of Science and of Medicine, and has also received many awards, including the Lesker Basic Medical Research Award in 2012. While he's well known for his discovery of kinesins, that you will hear more from his presentation today, he's also a very strong advocate for scientific education. He started the iBiology lectures aiming to make science understood, not only to the elite scientists, but to graduate students, and today to high school students. There are now, this really has inspired many budding young scientists to do science. There are now over 500 such videos available on YouTube, and it's also fully accessible in mainland China as well, where they have started their own program called iBioChina, and reposts many of the iBiology lectures with permission, using WeChat and other social medias. Professor Gibbons and Vales has made tremendous contribution by their discovery of microtubule-associated motor proteins, without which the process of cellular functional movement would not be possible. In recognition of their distinguished achievements and contribution, they are awarded of equal share this year's Shaw Prize in Life Science and Medicine. Without further delay, I invite Professor Val to the podium to first introduce the video of an interview with Professor Gibbons. He will then share with us his topic on molecular motors, the engine of life in person. Professor Val, please.
Well, thank you for that wonderful introduction. And it is my great honor to receive this year's Shaw Prize and to come here to the university to speak with everyone today. First, before I give my talk, I'd like to introduce this video, an interview with Dr. Gibbons. So as you've heard, one of the really wonderful things about this year's Shaw Prize, I think almost my feeling was really grateful that I received it myself, but I was also so happy that I could share this prize with Dr. Gibbons. He's a wonderful scientist, a wonderful human being, and someone who's really pursued scientific problems with passion and I would say real purity of running a small lab that has been very, very focused on getting to the answers of fundamental biological problems. And he's been a real role model for me in science and, as you've heard, a hero of mine. So, as you've heard, unfortunately, Dr. Gibbons cannot attend due to health reasons. I know he would have loved to be here right now speaking with all of you. But we have here an interview which was done through the iBiology project, which I'll tell you a little bit more later. But this interview was just released this week, so you can view it through iBiology.org, and you can share this video with friends that could not be here today. So without any further ado, I present Dr. Gibbons in this interview, and you can learn more about his pioneering work on dynein and understanding the fundamental mechanism of how cilia beat and how sperm can swim. These are fundamental types of motion in life, and this is what Dr. Gibbons discovered and pursued throughout his career. So we'll hear now the video.
So welcome. Today we're talking with Ian Gibbons, who nearly 50 years ago discovered the molecular motor protein dynein. So dynein is a force-generating ATPase that causes cilia and flagella to beat. It's also found in the cytoplasm of most eukaryotic cells, where it transports all kinds of different cargo, such as small membrane vesicles, the Golgi apparatus, the nucleus. It transports mRNAs and protein complexes. And in mitosis, it also plays an important role in constructing the mitotic spindle. So it's a very important molecule in cell biology. And, well, I've known Ian for many years. We've collaborated on several occasions. But I've never had a chance really to sit down and talk to you about how this whole story came about, how you went about discovering dynein. So, well, let's go back to when it all started, 1963, and maybe you can tell me about what captivated your interest in studying cilia and flagella in the first place.
I was working as an electron microscopist at Harvard in the biology labs, and I was collaborating on a project involving looking at flagella in the various flagellated protozoa. And one of the days we happened to take some pictures by accident that showed a set of cross sections of cilia and flagella and the transition between the basal body or centriole up into the shaft of the axoneme. And there were an enormous variety of fibrillar structures there that had never been described previously. And I felt that they must be doing something important. I knew that cilia and flagella were motile organisms. My supervisor at Cambridge, where I was getting my PhD, was John Bradfield, and he had actually written a paper on cilia and flagella mechanisms at that time. But then he left the field completely and went into finance. So I decided that when I saw these beautiful structures, I really wanted to know more about how they might work. There was so much structure there; it was far from obvious as to what it might do, how the different bits might interact.
So which of these structures did you think might be the force-generating entity, and then how did you actually go about figuring that out?
Well, I didn't get there all in one go. It took several stages. And one of the things was that after I'd spent a couple of years doing electron microscopy, I realized that one only got a rather limited distance looking at structure alone, because there was almost nothing known about the chemistry and not a great deal about the detailed motility. And so I decided to take on the biochemistry, try and go for it myself, though I had no experience in biochemistry. But I did have my wife, who was an experienced protein biochemist, working at John Edsall's laboratory at that time, and she certainly provided me with lots of help in getting started and knowing the important questions that I should be considering. So once I did that, then I decided that the Tetrahymena Cilia were an interesting organism from which to isolate the cilia, since one can grow them up by the pound and isolate them. Actually, I bought myself a cream separator to harvest them, and we did get maybe about a pound of Tetrahymena in each culture that we were using. So then, from that, I used the method for isolating the cilia that had been developed by Sir John Randall's lab in London, and used that to isolate the cilia. His people had tried to look at the proteins, but they hadn't gotten very far, because they found that the proteins appeared to be almost completely insoluble. I suspected that the problem had been that the isolated cilia, after they fractured from the cell, the ciliary membrane healed over the isolated tip, and so the cilium was completely enveloped by the membrane, and so the proteins couldn't get out. When I got these preparations of isolated cilia, then I decided that the first thing to do was to get rid of the membrane, so I applied a mild detergent, digitonin. That did a wonderful job of taking off the membranes, and also showed that the structure remained intact. All the internal bits of the ciliary apparatus remained intact. And very importantly also, the cilia retained their actinyl ATPase activity. Very little disappeared with the membrane. So we had the denuded actinyls. When I just put them into a concentrated solution of salt, the protein all completely dissolved. That showed that they were solid, that you had access to all the protein, but it wasn't what I wanted. I wanted to get some fractionation, and that I achieved by extracting them at low ionic strength with EDTA to completely remove any divalent cations. When I did that, I found I got a very interesting separation of the protein into two fractions, a soluble fraction and the residual pellet. And comparing those two structures in the electron microscope, I saw that the extraction with low salt EDTA had removed all the central apparatus, the spokes, the two central tubules, and in addition had removed the arms, the inner and outer arms that ran along the lengths of the doublet microtubules. When I looked at the solubility of protein and ATPase, I found that that this had indeed achieved a good separation, because the solute, that soluble fraction, contained about 80% of the total initial ATPase activity, but only had about 30% of the total protein. So I'd obtained something like a threefold purification just by doing this simple extraction. And so, since I had most of the actinyl ATPase present in the soluble fraction, obviously the thing to do was to look at it. And the only technique one had at the time, this was before gel electrophoresis, the only technique available was centrifugation. Started with an analytical centrifuge, and there were three peaks. One slow one at 4S, one at 14S, and one at 30S. When I ran the same material on a sucrose density gradient and assayed for ATPase activity, then I found that the ATPase activity was completely confined to the 14S and 30S fractions. The 4S had essentially none, though it was the largest fraction in terms of total protein. So then the question was, which of the various structures that had disappeared had the actinyl ATPase actually come from? And to find that out, I tried recombining. I took the soluble fraction, the pellet fraction, mixed them together in the presence of magnesium. And fortunately, I found that a major fraction of the ATPase activity that had been extracted in solution became rebound to the pellet fraction. Looking in the electron microscope, this showed that the structures that had reappeared when the ATPase activity became rebound were the two arms that stick out from each of the doublet tubules. So this was pretty strong evidence that the acylemal ATPase was indeed localized in these arms, the inner and outer arms on the doublet tubules.
So now you had this ATPase. You could show that you can add it back and it could reconstitute this really interesting structure between the two outer doublets. Next step, how did you come up with the name of this interesting ATPase?
Once we had the protein more purified, gave it the name dynine. Dynine, but the dyne was a unit of force, the only unit of force present when I went through school. Actually, Barbara was the one who suggested the name dynine. She suggested it one evening at home.
So at that time, how did you think it worked?
Well, there were two theories running. There were two possible mechanisms. One would be a mechanism involving localized contractions, that somehow the localized sections along the length of the microtubule might change the crystallized packing arrangement of subunits in a way similar to that which the bacterial flagellum does when it changes its shape from a left-handed helix to a right-handed helix under the influence of a chemotactic attractant. That would be one possible mechanism. Second possible mechanism was that the microtubules would slide relative to one another, and that this, when opposed by some resistance to bending, would cause the formation of a new bend. Difficulty with that mechanism was that to have bends of opposite directions at different positions along the length of the acyline meant that the individual microtubules run straight along the length of the acyline, but it meant that sliding has to be in opposite directions in order to generate bends of opposite directions at different positions, even where you're only thinking of a single microtubule. That seemed to be a major obstacle. And but I didn't take it any further. At that time, actually, if you look in the text of the paper, I was actually leaning toward a contractile hypothesis.
So then what did you do next? It was time to do an experiment. So what experiment discerned between these two mechanisms?
After we'd taken the formality of publishing the more purified protein and the name, which is in 1965, then I realized that we needed to get more into the motility. And the Tetrahymena Cilia were extremely inconvenient for doing that, because they're only 5 micrometers long, and although you can see them, you can see they're waggling vaguely when you look with a high-powered light microscope lens, but you can never measure accurately the parameters of waveforms. So that was when I decided that we should shift to sea urchin sperm flagella, because these are about 40 micrometers long, about 8 times as long as the Tetrahymena Cilia. Their motility had already been studied by Sir James Gray, who'd shown that they propagate waves along the length of the flagellum. But we didn't have a real good source of them in Cambridge, Massachusetts, where I was then. And so we waited, and an opportunity came up for me to move to Hawaii, University of Hawaii, and decided that would be an opportunity to switch completely to using sea urchin sperm, since the sea urchins in Hawaii were sexually ripe all year round. You would have material for working with them year round without the complications that happen if you tried to do something like this in California or on the East Coast. So we moved to Hawaii. And then we started studying the motility in more detail. We first measured the parameters of the bending waves of the live sperm. Then we took the membranes off the way we had been doing for the Tetrahymena Cilia, showed that they could be reactivated with ATP and propagated bending waves very nicely. Then the question was, what was the mechanical force actually going on inside the acyline? And Keith Summers, who had my graduate student at the time, had the idea of using trypsin to weaken the structure. He thought that might do something. I had earlier observed that with the Tetrahymena Cilia, they become swollen when you gave them ATP. They didn't slide, but they became swollen. So he tried to make this happen also with the sea urchin sperm flagella, but it was completely unsuccessful. They didn't do the same trick. So he tried treating them with a bit of trypsin to see if that would get them to do the same trick. And, well, it did, but it also did something much more dramatic, because they just seemed to disappear completely. If you took the demembranated isolated axonemes, treated them weakly with trypsin, and then diffused in ATP slowly from the edge of the microscope slide, as the front of ATP reached the axonemes, they just seemed to fade and disappear. So that something quite dramatic was going on, something that had not happened with the Tetrahymena Cilia.
And you wanted to look at that directly and observe it.
I decided to use light dark field microscopy to watch the disappearing of these flagellar axonemes. So Barbara and I went back one evening and we looked and we saw it, and we were amazed that it really slid apart. It was very dramatic, and we were really, that was really one of the high points, because it was obviously demonstrating a very fundamental mechanism in the interaction of a motor protein with the microtubule for the first time. And it was happening under our own eyes. One of the things we did was to get more evidence that the dynein was actually the motor driving the motility. The way we did that was by partially extracting the dynein arms. When we did that, we found that the beat frequency decreased proportionate to the fraction of dynein arms that had been removed. And then we could add this solubilized dynein back again, and the flagellar beat frequency increased back almost to its original level as the arms became rebound. That was really the confirming evidence, the last nail in the coffin, or whatever you want to call it.
So we've covered the isolation discovery of the ATPase, dynein, to the in vitro demonstration that dynein can slide microtubules, and that was the underlying principal mechanism that gave rise to cilia and flagellar beating. So maybe you can give us a quick tour of dynein after that.
So at that point we wanted to characterize it biochemically, which we did, and by that time Lemley gel electrophoresis had been discovered and developed, so we were able to start looking at the polypeptides that were present in our preparations, something that had not been possible when we did the original extraction of the dynein, because there were no gels available that were appropriate at that time. So that was one of the things. We worked up, we purified this ATPase, we started looking at its polypeptides, we eventually cloned and sequenced one of the polypeptides from the outer arm, and then after we cloned and sequenced it, we started looking at the different forms of it, because it turned out, as most people discovered with gel electrophoresis, preparations were always more complicated than you'd like to think. There are a lot more proteins present than one really liked to think. So we discovered that there were about 8 or 9 bands in the electrophoretic position of the dynein heavy chains. And so we isolated different ones of them, studied how many there were, and that led to realizing that the dynein family of the heavy chains alone consists of at least 9 subfamilies. And 2 of these correspond to the 2 cytoplasmic dyneins. The other 7 are axonemal proteins, and sometimes there's more than one representative of a family present at one time. So usual flagellar axonemes like we were using contained 13, actually, gene products. So we cloned and sequenced one of these genes, the heavy chain, the beta heavy chain O-atom dynein, cloned little bits of others. But of course, as time went by, other people joined in, and so by now there's a real elaborate tree showing the 9 subfamilies of the dynein heavy chain form independent subfamilies that were present even in the last common ancestor of all eukaryotes. In 1997, we wanted to retire from University of Hawaii, worked less than full time. So we moved to California, where our family, to be closer to our family, our children, and there I started working on the cytoplasmic microtubule binding domain of cytoplasmic dynein. And by taking just that separate small piece that constitutes the microtubule binding domain and its coil stalk, which is only about 5% of the total heavy chain of the mass of the whole dynein heavy chain, we were able to express it in bacteria, study its properties. We were able to show that by changing the register between the two strands of the coil stalk, we could modulate the affinity constant with which this expressed microtubule binding domain would bind to native cytoplasmic microtubules. Having got there and having this expressed a functional cytoplasmic microtubule binding domain, the next step obviously was to go on and crystallize it and to get a structure. Took a year, but Joan Garbarino in my lab did a wonderful job, and then helped especially with the crystallography. We joined Andrew Carter from your own lab. Together we managed to get a reasonable atomic level structure for the microtubule binding domain.
Are there any particular thoughts that you have, just looking back over this?
My general modus operandi, I think, has been to work with a technique for a while, and if it seems to be sticking, try climbing another side of the mountain. Change your technique and start afresh and approach it in a completely different way. I mean, and I think that's useful. I've certainly found it interesting. I don't know how well it will work today, where there are many more people in science and most problems and more popular competition might make it difficult for you to do that, but certainly it worked very well for me, and so I would encourage anyone else to try doing it if they have the opportunity. But the main thing is to have fun, enjoy what you're doing, and then with any kind of decent luck, you'll get a great result.
May I now invite Professor Val to come on stage to deliver his lecture entitled Molecular Motors, the Engines of Life. Professor Val, please.
Thank you very much. I know that probably a third of the audience, is this on? A third of the audience is our high school students, not on. Can you turn this mic on? Okay. I'll use this one. I think a third of the audience are high school students, and I know there are also full professors in the audience as well. Oh yeah, I thought I had it on. Oh. Battery change here. Anyway, I will try to make it so high school students can understand this talk, and maybe then the full professors might wonder how I ended up getting a Shaw Prize lecture. But I'd rather make it understandable to the high school students. So I'm going to tell you about molecular motors, and you heard about dynein from Ian Gibbons, and how dynein creates the beating of cilia and flagella. But there are other functions of molecular motors too that actually are very familiar to us thinking about motors that we have in a city. So in a city, you have lots of cargo that needs to be transported from one place to the other. And of course, there are engines that do that, and these engines carry these cargo trailers. But what I'll show you in this talk is if you dive down and look at what's happening inside of a cell, you will also see a transportation system. You will see roadways, which I'll describe. They're called microtubules. And on these roadways are various types of cargo, which are building blocks inside of the cell. And these building blocks also need to be transported, and they're transported by very small biological machines, which are called molecular motor proteins, that travel along these cellular roadways. The study of biological motility, for which the Shaw Prize was awarded this year, is in fact one of the oldest fields in the history of biology. Imagine you being Leeuwenhoek and viewing this whole microscopic world for the very first time. And what Leeuwenhoek saw were amazing things that were previously invisible to mankind. Just looking in a drop of pond water, he could see all kinds of moving objects, which are cells, and he commented that the motion of most of them in the water was so swift and so various, upwards and downwards and roundabout, that I admit that I could not but wonder at it. And I must say, when I look at the microscope, even now, I still am in awe and wonder about the motion that is created by biological systems. This is the movement of cells produced by the cilia that Ian Gibbons studied, and you heard about from the video. But also, a very long time ago, Bonaventura Corti also used a microscope. And when he looked at certain types of plant cells, he saw that there was motion inside of the cell itself. He saw motion of chloroplasts moving around inside of the cell. And these old scientists had very kind of dramatic language, which I wish we could still use in the scientific literature today. But I know that the phenomena that I announce is too strange to be believed at first. I saw two torrents inside each section. One of the torrents rose on one side and descended on the other constantly. And this not but once, but thousands of times, and for days and for even weeks. Very dramatic. But this is motion inside of a cell. And here's another type of motion that I think everyone is familiar with, and that is the motion of DNA when one cell divides into two. And here is the DNA in green. And what you'll see are, during cell division, that chromosomes are formed. These chromosomes align in the process of mitosis. And then the sister chromatids, which are equal copies of the DNA, physically separate from one another to equally divide the DNA. Again, this is a form of biological motility. So what I'll tell you about in this lecture is that just the way that we transport ourselves in cities, that the cell also uses a system of roadways and motor proteins, and I'll explain to you what they are in this lecture. But first, maybe also for the high school students, I'll tell you a little bit about how I became interested in science. I grew up in Hollywood, California, which is known for many things, but not for its science, perhaps. Although, when I came here, I felt very at home here. I traveled halfway around the world, and I discovered my roots just 2 days ago. My parents were in the entertainment industry. It was very interesting that Run Shaw, of course, was famous for a pioneer in the film industry in Asia, and that is the background of my family. My father was a screenwriter, and my mother was an actress. So, yeah, very glamorous from a different time. But they were also very interested in really all the great things that human civilization has produced, including science. My mother took me to science museums. My father took me to lectures when I was a little older in astrophysics, which he also very much enjoyed. And when I grew up, I certainly grew up in an interesting town, which was Hollywood. And this was my elementary school class. And this was me. This is what I looked like when I think I was in 6th grade here. But I had also other interesting classmates. None of them turned out to be scientists, but there were some other people in my class that maybe some of you may know. Maybe this is even too old for the high school students now, but this is Michael Jackson over here. And as I say, probably you will go away. There is only one thing you will remember from this lecture, and it will have nothing to do with motor proteins. But I still feel like it's better for you to remember one thing from the lecture than nothing. So anyway, this was my environment, but I decided that the entertainment industry was not for me. I could not dance as well as Michael Jackson, so I decided to choose another career. And I did a science fair project in high school, and I think the big turning point for me was I really did not like my biology class at all in high school because I had to memorize many, many things. But when I started to do my own project and I started to ask my own questions, which is really what science is, it's being curious, asking a question that no one knows the answer to, and then even when in high school trying to answer a small question, doesn't have to be a big one, but just even a small question that isn't known that you can try to work out yourself. That was a very wonderful experience for me, and I still love that experience. So I love having this job where my job is I'm being paid to be curious, ask questions, and try to find out answers. It's a really wonderful profession. But I got that, wonder of science and curiosity in high school. And actually, this high school project ended up getting selected with another group of kids to go to Washington, D.C. But now I'll fast forward to now as a graduate student, and 1984, working with Mike Sheets, Tom Rees, and Bruce Schnapp, and we were trying to solve a problem of the motor systems inside of cells. And I'm first going to tell you a little bit about the roadways, and then I'll tell you about the motors. So the roadways inside of the cell that I'll tell you about are called microtubules. And they are shown in green here, and they are these linear tracks that are made of a protein called tubulin that polymerizes like beads on a necklace. So they form these long, long filaments that run throughout the cell. And if you look at this, you could see that many of these tracks all originate at this point very close to the nucleus. This is something called a centrosome. But all the tracks start here. So I will use the term here downtown because this is in the center of the cell. This is where all the tracks originate. And then they branch out from here to the very periphery of the cell, which I'll call the suburbs. So these roadways are extending from the center to the periphery of the cell, just like roadways in a city. So what are these roadways carrying? Well, in our roadways, we have engines here, but these engines are carrying cargo containers that are full of consumer goods. And inside of the cell, the trailers of the cell many times are things that are called membrane vesicles. They are like balls of lipids, and on the surface of these vesicles and inside of them, there are various types of building blocks that the cell needs for survival. So there's lipids, there's proteins, and these proteins and lipids need to be delivered from places where they're made in the cell to other places where they're needed. So these tend to be the cargoes that travel along these particular tracks. Now, to study this system, we decided to use a particular cell because it has the most dramatic need for transport, and that is the nerve cell. And the reason is that the nerve cell is a very, very large cell. It has a cell body. This is where the nucleus is, but then it has this very long tube. This is where an electrical current also travels down to the very end of the nerve cell, which could be innervating a muscle. So the downtown, where the nucleus is, and the very end, which is the terminal, can be separated by a meter. So, for example, in your body, the nucleus and the cell body could be located in your spinal cord. That's the starting place of that nerve cell, and it then has this long tube that travels down inside of your nerve bundle, and then the very end may be in the suburbs. So I think this is very hard to relate to how big this is. So in this next slide, I'm trying to do an imaginary experiment here. Instead of the cell body being 30 microns, which is very hard to relate to, let's imagine that the cell body is as tall as I am. I'm not quite 1 meter, but I'm close, 2 meters. But let's say, so 1 meter is about, thank you, is about this big. And if we put this on our scale here, this 1 meter cell body that would be sitting here, the nerve terminal would be in Shenzhen. And the objects that would be traveling from this 1 meter cell body to Shenzhen would be about the size of marbles. And those are the membrane vesicles, and they would be taking 2 days to travel from here to Shenzhen. So that's the scale of transport that is going on, relative scale of transport going inside of your body all the time, delivering goods back and forth. So now I've told you about the roadways. Let me explain what the machines are, what the motors are. There are 2 main kinds of motors, and this is what the Shaw Prize was awarded for, was the discovery and understanding of these motors. Kinesin, which I'll tell you about, dynein was discovered by Ian Gibbons, and you heard about that. Now, the interesting thing about these motors is they're very complementary because they move in opposite directions along these tracks. So kinesin travels along the microtubule. The microtubule is an asymmetric polymer. It has distinct ends, and the kinesin effectively is moving in the direction of the suburbs. And dynein, on the other hand, is moving in the opposite direction towards downtown. So this is a system where these different motors can transport goods back and forth in opposite directions inside of cells. Now, I'll give you some analogy of what these molecular motors are in comparison to motors that we are more familiar with, which are car engines. And in many ways, the analogy is very appropriate, only the size is very different. So your car engine, if you open up the hood, may be about 1 meter in size. And the molecular motor part of kinesin is 10 to the 8th smaller, so 100 million times smaller than a car engine. It also uses a chemical fuel. So your car uses hydrocarbons, while these molecular motors use a special high-energy molecule of the cell, which is the main energy currency called adenosine triphosphate. The motors move at about a few millimeters per hour. And this sounds very slow. It is actually quite slow compared to a car engine. But if you actually consider the relative size of these motors and how fast they're traveling compared to their own length, you can see that these motors, molecular biological motors, are moving about 3 times faster than a car is moving on a freeway. So relative to their size, they're moving pretty fast. And they're also, if all of our car engines were as efficient as molecular motors, we wouldn't have as much problem with climate change, because they are much more efficient in converting chemical energy into useful motion. You heard about the discovery of dynein, so I'll tell you just very briefly about the discovery of kinesin in 1984 with my colleagues Mike Sheets, Tom Rees, and Bruce Schnapp. And to use this, we decided to look at the nervous system, but we chose the squid as a model system. First of all, most people are not willing to donate their axons. So we decided to use a model organism, which is the squid. But the squid was also very good because its axon is about 100 times the diameter of a human axon. And that made it much easier to actually dissect out of the squid and then study. And this is where a beautiful place where we caught the squid at the Marine Biological Laboratory at Woods Hole and did this work. Now, at the time, a group of investigators, Robert Allen, Scott Brady, and Ray Lassick, made this beautiful movie of transportation inside the squid giant axon. So when you used, at the time, a very new microscope called a video microscope, this is what you see inside the squid giant axon. These large things are mitochondria. Students probably learned about that in high school. And these very small objects here are these small cargo containers, these membrane vesicles that are moving up and down. Now, our approach to understand this system is when you're looking at this complicated system inside the squid giant axon, there are many, many molecules, different kinds of molecules inside of a cell. Cell is very complicated. It's, in fact, this is, if you take a car, these are all the parts that are inside of a car. Well, inside of a cell, there are actually many more parts. In any average human cell, there are probably 10,000 different components. So figuring out which component is really essential for the motility or not essential for the motility is not that easy. So even with a car, you find out, well, the seat is nice, but it's really not essential for the transport mechanism. So what our goal was to take a very complicated system, which was the cell, break it open so we could, first of all, study it inside of a test tube, and then sort out, just like this, which are the components that were critical for transport and which ones weren't. And we were aided with this. So this was, first of all, when we started taking apart the squid giant axon, you can now see these tracks, which we discovered were microtubules, and these cargoes moving along it. And we then found out that we could replace these natural cargoes with small plastic beads. And we could trick the biological system so that it thought that these plastic beads were cargo, and the motors would attach to them. So what you're seeing right here are small manufactured beads that are moving along these microtubules just like these natural cargoes. And I don't have time to explain everything about this, but because we could simplify it one step further from these complicated cargoes to plastic beads, allowed us to find what molecule was transporting these plastic beads. And that was done in a snowy winter in this very small town of Woods Hole, where there was nothing to do but work, which was very good. Even the one bar in town closed, so there was really nothing to do. So that's what I did in the winter. And Ian Gibbons described a protocol called gel electrophoresis, and this is a way of identifying proteins. But anyway, this was the protein. This band and these two other bands were the signature protein that was causing the movement that we saw in the squid giant axon. My mom helped to name it. She actually came up with the name with her Greek friend, kine, which means movement in Greek. That's how we came up with the name Kinesin. And Kinesin was effectively discovered 2 decades after Ian Gibbons first described dynein. So Ian and I then have spent much of our careers along with people in our lab trying to understand how they work. And I'll give you a very short tour of this. But originally I showed you that these beads move along these tracks. That's what I showed you. But here the motor is invisible. You can see the bead, but the motor is still not visible with the microscope. And it was my dream to be able to see the motor directly. And this happened when I was on sabbatical, Toshio Yanagita's lab in Japan, where we got rid of the bead and put a fluorescent dye on the motor protein and used a very sensitive microscope that could see the fluorescence from a single fluorescent dye attached to a single motor protein. And that's what this movement looks like. Each of these are individual molecules that are 100 millionth of a meter here moving along these microtubule tracks. And if you follow this motion and zero in on it at very high temporal resolution and spatial resolution, you can see that while the motion looks continuous here, if you analyze it very carefully, you can see that the motor is actually taking steps. As it's almost like walking across, stepping stones across a pond. It's binding to discrete landing spots on this microtubule track. And I'll describe this, but the way the motor does this is it uses this chemical fuel, which is ATP, and it hydrolyzes it. It breaks a bond between 2 of the phosphates. It then releases one of the phosphates to a derivative molecule ADP. The motor then gets rid of the ADP, and it starts the cycle again. And this is like one stroke of your piston in your car engine. And during this one cycle, the motor takes one step along the track. Now, we wanted to know how it would take this particular step. And to understand that, we had to go to much higher resolution to know the shape of the protein at atomic detail, which we did with Robert Flederick, John Kula, and Elena Sabline. And this is what the kinesin molecule looks like with its fuel sitting in a groove in the protein at atomic resolution. And what this image is capturing one state of the motor. But we know the motor has to change its shape during this cycle. Now, the analogy is, let's imagine trying to understand a horse. And if you just took this one snapshot of the horse and say, how does it move? You probably couldn't tell me how it moved. Of course, you all seen horses. But imagine you haven't seen a horse, and that's all you saw. You wouldn't understand how the horse moves. So you need to take multiple snapshots of the horse to understand what it's doing with its body and legs to propel itself. And we had to get multiple snapshots and understand this motor protein in the same way. And as a result of this, this is an animation, but this is built upon atomic detail of what we learned from this motor. It binds this ATP, and it has this little arm here that's almost like a judo expert that flips its arm, and it moves its partner motor domain from a rear side to a forward side. So the motor is made up of 2 identical motors, and they play leapfrog with one another. The motor in front essentially pushes the rear motor to a forward side, and it's like climbing up a rope with a hand-over-hand motion. So we actually understand the details of how it works. We then wanted to understand dynein, and this, one thing Ian didn't describe is how massive the dynein molecule is. It's like a giant Goliath compared here to the very puny little kinesin molecule. But it's been a great pleasure to participate in that journey with Ian. And Ian and I, this is actually a recent picture of Ian and I actually celebrating the Shaw Prize with Peter and Wendy. But we published several papers together. And some of the work that Ian described and was done by Ian, as well as some work done in our lab, gave rise to this model. Again, this is based upon atomic structures of dynein carrying a cargo, but it's a very different looking machine, and it moves in a very different kind of way. The kinesin is a very organized molecule where it's almost choreographed. The two molecules are choreographed. Here, the dynein molecule kind of executes almost a drunken walk along the microtubule surface, where the two heads actually act very independently of one another, but the changes in the shape of the motor propel this little blue device, which is the binding site for the track, to explore space, but propel itself from a rear site to a forward site. And sometimes the dynein actually moves backwards. It changes tracks. It has various kinds of motion to move forward along the track. So anyway, just to conclude, these motor proteins are essential for life. I showed you that they're necessary for the biology and the motility of single cells. And I haven't had time to go through all of the biology, but the swimming of sperm is what starts life, human life in the first place, to fertilize an egg. The way the single fertilized egg goes to two cells, that's the first step, next step of life. That fundamental process of cell division, again, involves molecular motors. And then the whole process of development, again, is a choreographed process where the right components need to be placed in the right part of cells and tissues, and many of these motions are done by molecular motors. So when you are born, you can be very grateful for all of the molecular motors that you have inside of you. In some cases, molecular motors, there are genetic diseases now that we know also occur in molecular motors and give rise to a variety of diseases. But by understanding molecular motors, there's also hope that we can actually develop potentially drugs that might be useful for treating some of these diseases. And again, it's been the work that I described to you is really work from many, many people, more than I can describe. These are many of the fantastic students, collaborators, colleagues that I've worked with over the years. This is my lab now, and one of the great things about being in this profession is I constantly get to work with young people all of the time. They go off and they get jobs, but then there's new young people who come in and take their place. So, and that's this is my lab right now. They're doing all kinds of interesting work. And again, just to advertise iBiology, this is a great resource. We may talk about it in the special session, but you can go to this site and, this is Randy Sheckman, who was involved with the Shaw Prize, but you can go and hear about 500 different talks by leading scientists if you're curious to know more. So anyway, with that, I thank you, and I thank you for the invitation to come here.
Thank you very much for your inspiring lecture, Professor Val. Please remain on stage. We will now proceed to the panel discussion and Q&A session. May I now invite our moderator, Professor Chen, to join Professor Val for the discussion. Professor Chen and Professor Val, please.
Thank you. So, thank you, Ron, for this amazing talk on your journey, or your scientific journey in the discovery of kinesin. Actually, it's more than just a journey. You've actually showed before that. I'm not so sure too many of us would show a photo of our grade 6 class without a famous individual in there. So this session is open for questions, and I would encourage young faculties, postdoc students to question first. Can someone run the microphone for me, please?
I have two very sort of random questions. One is on that really amazing video of you substituting the plastic beads for the vesicles. It seems like all the motion's on a two-dimensional plane, and I wonder how you managed to, get all the microtubules to line up on that two-dimensional plane, so that, or is it? I mean, obviously the cell is a three-dimensional structure. How come it all seems on that two-dimensional plane?
Yes, that's a good question. The two-dimensional plane is physically the glass cover slip. So
you're smushing it down to.
Focusing, it's just being focused on that glass cover slip. So there may be some microtubules floating in the solution
but
as well.
That network that you see, that
network is actually
smushed onto the.
Yeah, it actually sticks. So it's not actually being smushed because it's actually so small. It's the diameter of this microtubule is 1/1000th of an inch. So it's almost like even physically impossible to smush it. But there are ways in which you could add proteins to the glass cover slip to make it sticky for the microtubule. So the microtubules are actually sticking there.
That's fascinating. The other question, I was curious about the nature of collaboration. So now you employed many different tools on the way to investigate this protein. I'm just curious, so once you reach a certain point and you want to employ a new tool and you collaborate with another lab, how do you go about doing that? Do you, do you search out people who are experts in certain techniques and then write them out of the blue, or hi, I'm working on this. I need some help with this particular technique. Do you mind collaborating with me on, or is it do you meet them at conferences? And just curious how that works, because it seems like you've used many different tools in learning about this protein of yours.
Yeah.
Over the years.
No, it's a great question, and it's kind of all of the above. But the question is, when you do, I'm reframing this also for the high school students. When you do science, some of these problems are very difficult and very challenging, and it's sometimes hard to really do everything by yourself. And that means that you have to be also very smart to get other people who can help you if you don't know something, who can complement what you know to be able to solve the problem. And this is really important. First of all, it's important because it's much more enjoyable. I think many people think of scientists as being, really lonely and, antisocial, and they just, they're sitting there with their test tube all day on. That's kind of the way the popular press almost, frames a scientist as, kind of this lone person or lone genius. But it's like the farthest thing from the truth. Many of you are graduate students, so but the lab is like a family. It's very close. You're working with other people all the time. But also for getting a project done, many times strategically you have to pick people to work with that, first of all, as I say, can complement you. If you don't have a reagent or you need to know a technique, then it's really very important that you find someone who can work with to do that. And also partly it's good to pick personalities that you feel like you can work with as well. It's just human nature. Some people tend to work better together than others. But the bottom line is, for biology these days, collaboration is essential. Biology is getting more and more exciting, but it's a very fast-moving field. And it's almost an essential part, I think, of scientific training. Not just to learn techniques in the lab, but also really to strategically learn how to collaborate. It's a skill. And I would say that should be part of your graduate education, honestly, to learn how to do that. There are a lot of people that are worried, oh, you know, I won't get credit, I won't get, you know. The most difficult part about science is actually just not getting results. That's the most difficult part about science. It's not like losing all this credit. So, you know, my feeling is work with other people, because if your project is moving well and fast, then you're having fun, and then science becomes fun. And if you need to work with other people to help that project move, that's what you should do.
Thanks very much, Danny. Ron, I'm going to remember many things from your lecture, not just Michael Jackson, but the thing I'm particularly going to remember is...
Name one other thing.
The drunken walk. I'm going to remember the drunken walk. And what I wanted to ask you is about what controls directionality. Because you know, if you're a drunk, then you go all over the place. But they did seem to have some purpose to their drunken walk. But you mentioned that they sometimes go in the opposite direction. So what controls directionality? How does transport get driven from, let's say, the top of the cell body in the neuron all the way to the suburbs?
Yeah, so how does the directionality of dyning, in this case you're talking about dyning. So both of these motors have a mechanical element. So in the Kinesin, it was that arm that was connecting the two motor domains. And that arm would swing from kind of a rearward position to a forward position. And when it went forward, that's what pulled the other Kinesin head. So there, I didn't really point it out, but there is a mechanical arm in dyning too. The dyning has a ring, and it has a big part of the motor protein that kind of arcs over the top of the ring. And that undergoes a change from a straight state to a bent state. And when it goes from the bent to a straight state, it also swings that long kind of arm, which at the very end has this microtubule binding domain. So it basically kind of throws it forward in the right direction. But it doesn't exactly land it in any particular spot. So after it throws it in the right direction, it's kind of searching around for where to land. And that's what that searching process, which is a phenomena called Brownian motion or thermal-driven motion, is also what is causing that kind of randomness of where it is landing. And sometimes that random thermal motion actually causes it to bind in the wrong spot and even walk backwards. And you can see in the dyning traces, sometimes it's moving forward, sometimes it takes a step back, and other times it's moving forward. So in reality, and this is also useful to know for the high school students as well, what life is like as a molecule in a cell is an incredibly vigorous environment that's very hard even to imagine for us. Like if all of us were molecules right now in a cell, and this room was a cell, we would be bouncing, we would not be sitting down. Because when you're that small, water molecules are constantly hitting you, and you're moving around constantly. And that randomness is what these motors are trying to fight against. So they're in fact working in this incredible environment of random motility and trying to create order out of it. And so dyning is kind of against this, battle of, trying to get this microtubule binding domain to land in the right direction, but it's fighting against this thermal motion. Why dynein is not as efficient as kinesin, it's a mystery. One idea is that, kinesin is running along the bottom part of, in this very purposeful walk along the kind of right next to the microtubule surface, while dynein is kind of this big giant trying to land on top. But it may create an environment where the two motors can pass one another easily. So that may be an advantage. So sometimes that, you can imagine if two motors were on the same lane of the freeway and they're running into one another, it's not going to be a happy ending. But because the microtubule actually has like 13 lanes. It's like a 13-lane roadway. Maybe because the dynein has this more random motion and can move around the microtubule surface, it can dodge around the kinesin molecules and they don't collide, which is actually what you see in cells. You can watch organelles pass one another on a single microtubule, and it looks like they go straight past one another without colliding. So maybe that was part of the design of why, dynein is a little more drunken than kinesin.
Thank you for the fantastic demonstration. I want to ask two questions. The first question is, it goes from the downtown to suburbs, and in Hong Kong there are a lot of roadblocks. And it doesn't go straight and with one speed. Down the whole nerve cell, would it be faster around the downtown or faster in the suburbs or in the middle? The first question. That's the question.
Yeah. No,
why? The second question. Sorry, let me go finish. I see that you're talking about cells, right? Inside the cell is so organized, but yet I believe outside the cell, the extra cellular matrix, whatever it means, is not as people depicted as kind of random. It's probably organized, not as much as a cell, but organized. What it meant, I want to just simply ask a question. Talking about motors of the life, that's dyneins and kinesins found in outside the cells.
Yeah, both good questions. There are no known motors outside of cells. So the outside of cells seems to be made of molecules that have different sticky properties, and the cell kind of is testing its environment by how sticky it is, and that's often how neurons, a nerve cell has to find its way from the spinal cord to the foot during development, and it's actually testing its environment, mostly by its stickiness, its interaction, and not by motility. The other question you asked is, how smooth is it, or does it run into roadblocks? The motor definitely does not travel along one microtubule. The one microtubule, first of all, isn't long enough to go all the way from one end of the cell to the other. But the inside of this axon is packed with many microtubules. So if you fall off of one roadway, there's another one close by. But the other interesting thing, I think, again, for students, a lot of times we think about the inside of the cell as like a little bit like what I showed you there. It's almost like water. Really, the inside of the cell is like a jungle. It is absolutely packed with molecules. And if you look at it by electron microscope, there's almost no free space there. And it's actually remarkable how anything moves inside the cell at all. So the important point there is that the motors also are producing force. They're actually quite strong, and they're able to drag these big organelles through this jungle. It's almost like tearing through this jungle and ripping apart branches as you're traveling through. That's kind of more an appropriate analogy of what's actually the inside of the cell looks like again. It's like tearing through a thick jungle. What about high school students?
Yes, question from high school students.
Professor Val, thank you very much. Very enlightening lecture. I'm C.K. Chen. I am an anesthesiologist. Fascinating. I want to ask you, now you get a lot of people helping you. Just how do you get the funding? Is the university pays for the funding, or just give us some highlight, how do you fund the research?
Well, I guess I am very lucky. So I am not a typical, maybe average scientist in the U.S. system, because I am funded by a medical institute, the Howard Hughes Medical Institute, and they fund maybe like 350 scientists in the U.S., and it provides most of the funding just from that one source for all of the research. So I should acknowledge them, but I also have a smaller grant from the National Institute of Health. So no, I've been very fortunate to have grants from both of these funding agencies. And, as I said in the Shaw Prize as well, we have to be grateful to the public, because it's ultimately the public that is supporting the National Institute of Health in the United States and our research. So I'm sure here as well. So we have to, as scientists, really make sure that the public and the government also really understands the importance of the science that we do.
Maybe I'll follow up with this question, Ron.
The high school students are leaving because they are afraid they don't want to ask. They don't want to ask a question.
Oh, no. They go home to do their homework. Because I think
there's
a lot. Well, is there a question from the high school students? Yes, there is one.
It was very inspiring to hear about your scientific journey. I was just wondering if you have any advice to anyone looking into going into academia as a career or into scientific research. What sort of advice would you give to a university student looking into?
Do you hear that?
Yeah, the question is, what advice, do you have for someone pursuing an academic career? Well, I would just say, it's all about, it's not easy, but it's all about passion. I mean, you say, okay, if you, in fact, I think in many ways in science we're much luckier than a lot of other very creative professions. I mean, honestly, I watched my father, it was not easy to be a writer. And it's not easy to be an artist. It's not easy to be a musician. But I think it has some of the same elements of creativity and passion. I think if you love it, if science is really what you love, then just go for it. I mean, that's my advice. Just like I would say someone who's very talented at art or music or whatever. But, I mean, in reality, it's actually, fortunately, much easier to get a job in academics than it is in, a lot of the art disciplines. But it's still, it's very possible to have a great career in academics, and I love it. I mean, I couldn't imagine any better job.
Professor Jean.
So there are still some PhD students, postgraduate students. I mean, the high school students are gone. So I know that you have written a lot about how PhD education should be. So could you briefly tell us some of your major concerns and advice?
Oh, about PhD education? Wow, that's a big question. Yeah, well, I mean, as a word for iBiology, we're actually starting a course. Is it next week? We actually, so iBiology is this project to, have leading scientists describe their work, but we're also starting to take on some of these topics, like how do we best train PhD students. So actually in a week, there's a course you could take. It's called Planning Your Scientific Journey, which is a little bit about very important issues, which is how do you pick a project in science? How do you plan how to execute the project? I think those are issues that we're trying to do in the course that I think we often don't do a great job teaching our students. You know, in reality, there are two elements about being a scientist. One is information. So, you know, we have courses, you learn about gene regulation, you learn about the microtubule or whatever. So we're very good as a profession about, like, conveying information. We're not so good as a profession about conveying the apprenticeship of being a scientist. And I think a lot of PhD students learn this the hard way, meaning that we don't really explain to them, you know, a lot of the fundamentals of that we've learned, you know, from being a scientist for decades. But you know, sometimes students have to learn that the hard way by, often going from this transition where they've been consumers of knowledge all their lives, right? You know, middle school, high school, college, you know, consumers of knowledge. The transition to being a producer of knowledge is a fundamentally different way of thinking, different way of training, different way of being. And it's very exciting, but it's sometimes a shock for students who also have been incredibly good at school, to go to this transition of how to actually learn information that no one else knows and how to go about doing that. And honestly, I don't think we do as good a job training students as we can in how to make that transition.
But I think actually, in addition to that, Ron, the research environment from the time that we started our career to now is really quite different. There's funding that was talked about, and also the pressure to publish. And in addition, we are always asked to find a translational value to our research findings. So how does the young scientist adjust to this sort of value-driven approach in research and yet satisfy their curiosity, sort of blue sky research, that balance that we know that there's a need that's so important for new discoveries in science?
That's a great question too. Well, I have a opinion on that as well. But I think in part it's our job as senior scientists to, I think, present the reality of what the scientific career is. You know, as like it's not trivial to get funding. I mean, you have to have a really good project, a really good grant. Not everyone gets funded. Not everyone succeeds in science. Not everyone succeeds at that transition, as I said, to being a consumer to a producer of knowledge. Not everyone succeeds at that. However, I do think it's our job as senior scientists also to offer a protective environment for young scientists and graduate students to be creative and to be freed of those worries. Because honestly, what I see is so many young people almost going the other way and being like almost having unrealistic worries about the scientific profession. And I almost have to like explain to them, well, like, calm down. It doesn't matter if you don't get your paper in Nature. Life will still go on. I think we as senior scientists have to, live that battle of writing grants, getting that funded. But that is not the job of graduate student, right? Their job is to learn how to be creative, learn how to be curious, learn how to ask a great question, which is not easy. Learn how to ask a great question and solve it. And it's our job to help teach students how to develop all those skills. And you don't need to get some high fancy paper, to succeed, especially as a graduate student. I think if you get a decent paper out as a graduate student, you can still get go on and get a good postdoc. So I think it's a very important time as a graduate student to learn if you have the passion for science. That is a question for every graduate student that you have to ask yourself whether you do have that passion or not. And that's something that some people find they have and other people find they don't, and if they don't, they can go on to other careers. But I think they need to figure out whether they have that passion or not in a little bit of a protective environment where they're not constantly worried and they can learn how to be creative.
Okay, thank you, Ron. I think we've gone way over time, so I think there's so many different questions. So I really like to take this opportunity to thank you for addressing all the questions and your advice on scientific education and sharing your knowledge and wisdom, and allow us to take part in your celebration of winning the 2017 Shaw Prize in Life Science and Medicines. Thank you very much indeed.
Thank you, professors. Let's again give a big hand to the Shaw laureates for this exciting lecture and discussion. Thank you, professors and all, for joining us today. So this is the end of the Shaw Prize lecture, and I wish you all a nice evening.